Imagine a world where we can do computation inside living cells.
The problem that we're trying to solve is really trying to have a more sophisticated diagnosis that can happen automatically inside cells.
Imagine a biological computer that operates inside a living cell. So for example, it could be used to determine whether a cell is cancerous, and if so, then trigger the death of the cell.
So here we're talking about little molecular systems that you can, that run in a test tube or maybe even in a live cell, so they're really small. The type of work that they're doing is essentially they're trying to sense, analyze and control molecular information.
In this project, we're trying to use DNA as a programmable material.
DNA is highly programmable, just like a computer, and we can program a whole range of complex behaviors using DNA molecules.
So we're taking advantage of a phenomenon called DNA strand displacement.
So DNA strand displacement is essentially a competitive hybridization reaction. So it's two strands of DNA that come together, and as they bind to one another, a third strand that was initially bound is kicked off. One issue with any biology research or biomolecular research is that it's always sort of a cycle of trial and error. You test it, it doesn't work, so you go back to the drawing board and you do that over and over again. And it's a slow cycle because doing experiments just is hard and it takes a lot of time.
We've developed a language for programming molecular circuits made of DNA. So the programmer would write down a collection of DNA strands, and the software will work out how these DNA strands interact with each other and can be used to predict their behavior over time. And this kind of software could, for example, be used to detect and fix bugs in a molecular circuit design before that circuit is built. For decades, biologists had been using chemical reaction networks as a means of describing the behavior of biological systems.